topic on status of Trichoderma as a biopesticides with special reference to northeastern India. And I'm really glad that Professor Awasthi has agreed to give such talks. As we all know that agriculture is the backbones of our economy, and farmers everywhere face several challenges towards either soil degradations, pest management, resistance, those all raising the cost what we are using as chemical pesticides. In this context, biological solutions is always welcomes, and trichoderma is offers a sustainable path towards this. Trichoderma is a fungi which is widely known in the soil and plant roots, and they have the ability to suppress the plant pathogens through competitions and other parasitisms. They produce several enzymes that degrades towards our harmful pathogenic plant fungi. They also stimulate many times the plant growth by enhancing nutrient uptake. So we have a very good expert on this. So before introducing him, I must also tell you about MBSI, which has been initially been be started with Professor Dr. Aravind Deshmukh sir, who is our president's working. He has a very vast knowledge. In his absence here, he has some important work. Otherwise, he was promised to join on this. And during the course of time, he messaged me that he will be again joined to us. So he's leading, and we have many promotional scientific activities. And I, myself, Dr. Sujoy Kumar Panda, working as an associate professor at centers, along with my team members, my colleagues, Dr. Sivani Mahapatra, we are leading towards promotions of biofertilizer and biofertilizers throughout the countries. So we are committed towards the scientific knowledge, collaborations, and wish to be bring by doing several talks and bringing the new science towards on this. So now I have pleasure to introduce our today's speakers, Dr. Durga Prasad Aswati. He did his PhD in plant pathology and now serving as a senior assistant professor at Department of Plant Pathology in College of Agriculture in Tripura. The place where he comes itself, his work on Northeast is really very much interesting. And these are actually the states where actually organic farming has been very commonly used and has developed many of these. So he has been actively involving not only in the research, also teaching towards agriculture students. His research include vast areas as he is specialized in plant pathology, mycology, biological controls, mostly dealing with trichoderma as a disease diagnosis and management. He has worked with this integrated disease management part of IPMs, post-harvest awareness, and everything towards sustainable crop protections. He has enormous contributions towards agriculture in controlling several crops and vegetables, not only like a dozen of when I read of his publications on there, and mostly dealing with the fungal infections like Lagidisplodia and Rhizoctenia solani, Sclerotinia rolfosi. There are so much of other pathogens also he has worked. And his most inventions I found is including always towards disease management using either Pseudomonas or Trichoderma as a genus levels on there. He has also supported by ICR research programs. He is also doing promotions towards biofertilizers, biopesticide, working a lot towards climate resistance crop productions in northeastern regions. Several of his papers, at least more than 50 to 60 peer-reviewed papers, is published in international reports. He has several professional interests emphasizing towards sustainable agriculture, biological crop protections, effective utilization of microbial resources for improving crop health and productivity. So it's not all of the about. I tried from his CV of them to let you know and introduce him. Sir, thank you so much. And we really grateful for you to give your talk and today's to be participating and giving towards our students is and others as a lecture. May I request you, sir, kindly to go and present your topic. Thank you, sir.
Thank you, sir. Thank you. Am I audible?
Yes, sir, you are audible.
Okay. Okay. First of all, my heartfelt thanks to Microbiologist Society India and especially to Dr. Sujogya Kumar Pandaji and Chairman, sir, to providing me the opportunity to present my work, and Dr. Shivani Mahapatra, uh, who is continuously uh, have been associated with me for this program. So, first of all, a very warm welcome to everybody, the professors, faculty members, scientists, students, research scholars in this awareness talk series. So, first of all, actually, I want to thank the Microbiologist Society that they have chosen this topic. Is my screen is also visible, sir?
Yes, sir. But you can change one so that we can tell you that yes, it is
Yeah, yeah,
changing.
yeah, yeah. Okay,
Yes, sir, it's moving, sir. Everything is
okay,
okay.
okay then. So, uh, the topic was that topic which we have given. I have been given is status of trichoderma as a biopesticide with special reference to Northeast India. Here, sir was also mentioning, uh, Panda sir was also mentioning it that especially in Northeast, the scope of trichoderma is very high because we have a very vast divergent regarding to biodiversity. We have a huge biodiversity as well as our the biodiversity of both the hosts as well as bioagents as well as pathogens. So everything will be there. And the environment of Northeast is also suitable for actually bioagents like trichoderma. So that's why in India we have this type of climate a little bit in Kerala also. So the climatic condition which are prevailing in our Tripura or Assam part is little bit similar what I have observed in Kerala because both these states receives high amount of rainfall. The sand is a little bit hilly sand, reddish, and in this northeast, if we will just in the Shivalik range, so if we will not consider the upper Himalayas or the middle Himalayas parts, in northeast the 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 area is like that, that there will be heavy rainfall, evergreen plantations. So the bioagents are also favor. It's very easy to grow bioagents as well as to this one. Their bioefficacy is also very high. Now, the topic, first of all, this biofertilizer and biopesticides. So, trichoderma is a very important biopesticides, which is very much popular among plant pathologists, among microbiologists, and many other persons who are working in the field of agriculture. So, first of all, I will start about its introduction. So, every one of us actually we are always listening that trichoderma, the word trichoderma. So it is actually Greek word. It is derived from Greek, and here trichos means hair, and derma means skin. So trichoderma is a word which is derived from a Greek, and here the it means a hairy skin fungus. So we know that it is a very good and potential biocontrol agent in agriculture. Uh, at present, if we observe that, we will find that there are around 200 commercial biopesticides available throughout the world of Trichoderma. It can be different continents of world like America, Europe, Asia, and Africa. In India also, if we will we will go to the market, we will find that Trichoderma is dominating the in the shops of agricultural shops who are selling the fertilizers, biopesticides, or even insecticides, fungicides. So on those dealers, distributors, you will find among biopesticide, trichoderma is a major one. To name a few, I can say that PCI, we all of us we know Pesticide Control India. So are selling. Then there are many other also, but uh, but yeah, definitely the product before buying, farmers should see the product that the labeling is there and it is not expired, all those things. Now, the how we will isolate it. So actually, from soil only, we can isolate this trichoderma. Everybody knows. Just I am telling about, maybe there are some students also in this group. So for the benefit of them, I am telling the basic things. Otherwise, it is being known that it is a soil fungi and we can isolate it from soil. But the thing is, the beauty is that it is available in almost every type of soils, like loam soil, sandy soil, clay soil. We can find trichoderma in climatic conditions where there are little bit temperature is high, that we can get strains which can survive in high temperature, and in areas where the temperature is optimum or low. So scientists and ICR as well as CSIR, other institutes, they are actually, they have done enough work they have done to isolate the potential isolates of trichoderma. However, the we know the situation is changing because the native isolates of an area may have some more potentialities, and maybe those isolates have not been discovered or have not been found. So actually all of us should carry out the basic primary thing to isolate the organisms. So the Trichoderma harzianum, viridi, and asperellum has been registered for use, and we may find these organisms. So so if we will see the history of the thing, we will find that this genus was first proposed by Persoon in 1794 in Germany. And in 1865, the Tulasny brothers in France, actually, they illustrated that there is a link between Trichoderma viridi and its teleomorphic stage, that is sexual stage, Hypocrea rufi. uh, Re- Refi excluded, according to him, the Gliocladium virens is uh, is not the same as Trichoderma species. So later on, it has been proved that Gliocladium virens is now it is named as Trichoderma virens, and it is very much the publications has been given that this bioagent produces some compounds like gliotoxin and viridin. So this is the fungus actually of hypocrea. The picture of hypocrea, that is your teleomorphic stage of the bioagent. Now in nature, actually, when we will go, we will not find the asexual stage is more dominant. So we will found the asexual stage uh, of trichoderma, and teleomorphic stage is less found from the soil. However, the asexual stage of the fungus is widespread around the different geographic locations. As we have already seen that, see here, it is grown almost in America, in the America, Europe, Asia, Africa, all these continents are actually, they are producing this fungi as a biopesticide. So this is the thing where we are getting the link. Now, it is a dominant component of microbiome or among fungi. It is a dominant in soil ecosystem and almost in about all climatic zones. The peoples who are from Odisha also, in Odisha, I know it's a very means a different type of climatic conditions prevails. And in Odisha also we have a very good jungles and all those things. So forest area, so from there, the students or research scholars, you can try to isolate the organism. So the potentiality of trichoderma as a biological plant protection was first described in 1930. So it has been observed that at that time the scientists found that Trichoderma lignorum can protect citrus seedlings from the pathogen Rhizoctonia solani. So that was the first earlier these reports of potentiality of trichoderma as a bioagent. So it is more now around, I think, more than 100 years. So it has been actually lots of work has been done, and it's now well established thing. The biocontrol properties of trichoderma has been studied extensively, and the soil-borne pathogens mainly, like in agriculture, we know that there are the pathogens which survive in soil. So, like Sclerotium rauxi, Rhizoctonia solani, there can be the Phytophthora also, Pythium, then some other fungi like Verticillium. So this is very much effective among the pathogens which are soil-borne. Now, this is the what some classification of the trichoderma. So all the kingdom, subkingdom, subclasses, all things are given here. Now, as a fungal antagonist, uh, we know it is a non-pathogenic fungus. The majority of them, however, little one species is there which is found to be a little bit pathogenic to somewhat to other organisms, but more or less 99% it is non-pathogenic, and it is a biocontrol agent, and it suppress or kills the harmful plant pathogens. Mainly, it is very effective for managing the fungal as well as few bacterial pathogens. So, apart from trichoderma, actually there are many other fungal antagonists which we can use in plant disease management, like Gliocladium, Ampelomyces. The list is given here. Candida, Pacillomyces. So the species is different. Pythium, although it is a pathogen, but here species is different. Then Verticillium lacani, we know that we uses it in to control insects like Beauveria. It is not mentioned here. Beauveria, we can use Metarhizium. We can use for managing different diseases as well as for managing different insects also and nematodes also. Now, now here again, to the host range, Trichoderma has a very vast host range of different fungi. Gliocladium also, then Coniotherium. It has a host range of Sclerotinia. Then there are Pythium none, which can manage Pythium vexans and ultimum, which is actually a plant pathogen. So, like that, Ampelomyces we can use for powdery mildew fungi, and there we can also use non-pathogenic strains of fusarium. So because this normally it is a pathogen, so we have to find it out. If there is some non-pathogenic, then we can use as a source of biogen. But among them, the most popular for fungus is trichoderma definitely, and for bacteria it's a pseudomonas as well as bacillus. So, as we were discussing, that it can target a large group of pathogenic fungi like Rhizoctonia, Fusarium, Sclerotium, and Verticillium, and many more. The lists are very high, so I am not actually naming all of them. But yeah, definitely, if we will go to farmers' field, we will find that these pathogens are very much dominating in Northeast India. So wherever we are going, we will find the Fusarium in the soil, the sclerotium, the rhizotonia, and verticillium. Then some oomycetes fungi also, like pythium and phytophthora, which are causing damping of root rot and other diseases. So there are some specificity also. When I was searching the literature, I got that Trichoderma stromaticium can control witch's brew. So it is very much certain species might target more effectively specific pathogens. So it targets a specific pathogen that is Monilioptera, which is causing witch's broom in cacao. So this is the symptom. So here we can observe the symptom. So this reference shows that there are some strains which may be a little bit specific also to manage some effectively to manage some mm, fungi, fungi which are pathogenic. So, why actually we are getting the trichoderma is so popular? So actually, the thing is that it is commercially available and it is produced in India and marketed in India and with a grand success. I hope that for other bio agents also in future, government will have policies and we will be able to market them because we all of us we know the harmful effect of pesticides and which it is causing to our ecosystem as well as to our body. So let's hope that this popularity of trichoderma will become more and more. It will rise more and more with other bioagents also, which we have just seen few slides back. They will also be popularized in India. So there are many species of trichoderma, the many. I have only chosen five or six here. The Trichoderma harzianum, Trichoderma viridi, asperellum, Trichoderma virens, atroviti, and meticum. So actually, I have whatever we were previously the mycologist, we were only using the microscopic view and the development, their cultural and morphological um, observations were taken to classify them. Now, nowadays, this molecular, with the help of molecular diagnosis, we can manage them, we can make dendrograms, and we can, the more detailed analysis of the things has been done. But before going to the molecular level, we should have the biotechnologists and microbiologists, they all agree that the basic study of cultural as well as morphological characters should be done there. Then only we should go for isolation of DNA and doing ITS and or beta globulin test to identify a organism in its molecular level. So the primary step for every one of us should be to identify them in your cultural plate to should have an idea. So the Hargianum, it is a this bioagent which is very much effective against Fusarium, Pythium, Rhizostoria, and it degrades sclerotia and enhances crop root growth. The viridi, there has been report that it's a seed and soil treatment suppresses damping of diseases. It not only suppresses damping of diseases, it also suppresses root rots, collar rots in cotton, tomato, legumes, and other crops. And it also secretes the all trichoderma this genesis. They secrete some antibiotics or chemicals, and like trichoderma asperella, it colonizes the root system and stimulates ISR. It's not like that Hargianum will not colonize, it will not stimulate SIR. Based on some papers actually, we are writing it like that. And it promotes robust plant development, and it is an effective against Oomycetes pathogen. Then Trichoderma virens, it all produces secondary metabolites, gliotoxins, and is a strong biocontrol agent against many diseases like damping of root rot, others. Trichoderma atroviridi, it produces volatile organic compounds, so it will also, this volatile organic compound will again induce resistance among plants and against pathogens, and it inhibits phytopathogenic fungi like Rhizotonia, sclerotium, and others. So now stromaticium, we have already discussed that it has some, specifically it has been included in IPM program, and it is active against to control witches broom disease in cacao plantations. Now, if we will say the Trichoderma harzianum, we will find that it's the structure of conidiophores, conidia, and our this phyllids, all this, it looks like a pyramid. It's like a pyramid. See, it's a like a pyramid type the things, and branching is there with a main axis will be their main axis, and there will be some lateral branches. These the conidia are subglobose to ovate and smooth walled. In Hargenium, it's a smooth walled, and the measurement is given here. While in Viridi, the conidia is distinctly rough. It looks like a rough and have vaulted appearance. The conidiophores here, irrespective of Hargenium, it has less regularly pyramid. So the pyramidal structure is not that much developed here, and it has short phyllids. So see, the previously by seeing in observation, observing in microscopes, micrologists used to distinguish. Then we were using it against different pathogens. So now if we will serve for virens, see, it's a like penicillin growth. So penicillin like, just we all of us, we have seen penicillium. So it looks like a brush type. And the conidia are ellipsoid to oblong, smooth walled here also. In hematum, Trichoderma hematum, it's the short, there is short branches for phyllids. They are like basidium type, and short, they are broad. See here, it's a short and broad basidium type phyllids, which are we can say flask shaped. They are little bit flask shaped, and conidia are oblong to ellipsoid. So these differences are based on microscopic studies only. So after that, actually, we need to go for molecular identification, and definitely, as we have discussed, that we have to isolate the pathogen or isolate the antagonist from the soil, and then we have to for isolation, every one of us know we have to go for serial plate dilution. We will be we will discuss about that. So let's see about its mode of action of trichoderma. So mode of action here, actually, it's we know that there are 4, 5 mode of actions. This mycoparasitism, antibiosis, competition, induced resistance, cross protection, hypovirulence, all are there. Competition, we know that competition for food. So actually, to farmers also, we advocate them that make your soil more having more good friends. Your soil should have beneficial microorganisms, not harmful microorganisms for crop. They should have beneficial microorganisms which will promote the growth of your plants. So, if the population of those organisms which promote the growth of plant are more, then in competition, the pathogens will lose, and the plant will win, and we will get more yield. The same concept to enrich the soil, I think it's my own idea that actually the same thing we are using in natural farming as well as in organic farming, where we are actually promoting good soil health by increasing the the population of microorganisms in the soil. So if this is another thing, the trichoderma, how it is helping to achieve better yield by these mechanisms, competition, antibiotics, and all. So this mycoparasitism, actually, myco is fungus, and when a fungus parasitize another fungus, is this mycoparasitism. There are some recognition factors in mycoparasitisms which actually have biochemical and which provide actually some biochemical and molecular signals to the mycoparasite to locate the pathogenic fungi and initiate an attack. So these are some name of some recognition factors like lectins, hydrolytic enzymes, beta 1,3 glucanase, chitinase, cellulase, protease, and lipase. So this it gives us idea that they produces these enzymes and they help to control it. See, these are the here the how I have found a very much similarity between, actually this is a dodder plant which is actually parasitizing another plant. So the in we tell it Sharnalata in Bengali, maybe I don't know in Odia language, but the thing is Cuscuta or dodder. So the mechanism by which it penetrates, it's the things are very much similar here also. So they will produce haustoria and they will derive the nutrition. So from the pathogenic fungi. So it will coil, it will enter into the system, then it will derive the nutrition and it will grow. So this is about antibiotics. So many volatile as well as non-volatile antibiotics. So this is interesting fact. People think that people normally have the idea that only non-volatile the antibiotics will be working, but in trichoderma, the volatile antibiotics are also working, and I have also experienced it in my laboratory. So there are certain species like Viridia and Hargenia that produces 6-pentyl pyro, so 6 pp. So this unsaturated lactone gives a distinct coconut-like aroma. Means if we will grow the fungi in, suppose in potato dextrose broth, you are growing this bioagent, then when you will extract, because when we will go for mass production, actually we have to grow the trichoderma on potato dextrose broth, and we have to extract the mycelium. After extracting the mycelium, the liquid or the broth will have a coconut like a very pleasant coconut like aroma. So every mainly the students, you can grow this trichoderma and you can extract the this mycelium, and then you can smell the broth and you will find a coconut flavor in the broth. It's very much interesting, and I think I don't know how many of us we have tried. You can grow this organism, and you can, if you have the correct bioagent which I have mentioned, you may get this uh, uh, the property smelling like coconut. So this is also an antibiotic, and it also induces resistance in plant. So it's a volatile example of induction of resistance. It is we also tell that it's a opportunistic organism and a virulent plant symbionts, trichoderma. So these fungi, they can invade first few layers of the root. So it can invade. We can actually seedling treatment. In seedling treatment, we exposes the roots of the plant with trichoderma. So this trichoderma may colonize a little bit the roots, hairs, and while that it will stimulate resistance in host against the pathogens. So by this, actually, it is has the ability to induce resistance among fungal parasites. So this is a very by chemicals, by the use of chemical, by the use of bioagents, we can induce resistance, and this mechanism of resistance is becoming more and more popular, as we don't want to kill all beneficial organisms to gain yield. So we need to induce resistance among plants so that the plants will take care of their own and the biodiversity is not being damaged, ecosystem is being saved, and our health will also be good. So we have to think about induction of, there can be induction of resistance by use of chemicals as well as by bioagents. So all these things now which we learn in agriculture like seed priming by bioagent and chemicals, they also induces resistance. So you can use trichoderma as a seed priming also to induce resistance. So now there are many diseases actually. What to say? But to name a few only in vegetables, it is very important. Vegetable crops or fruits as well as vegetables in horticulture, because in Northeast, horticulture is very important, very very important. Although in agriculture also we uses the bioagent, but if you keep the eye on horticulture, we find that we can control damping of on vegetables, mainly the solanaceous crops. They like tomato, potato, brinjal, they are being suffered by damping off caused by Pythium, uh, ultimum, Aphanigvermatum, or some other organisms like Fusarium. And there can be a rotting of vegetables and flowers, even especially the post-harvest fruit spices are also grown in Northeast India. You may find may different kinds of leaf spots, even in oil seeds as well as in pulses also. We find leaf spots. So we can try to manage the leaf spots here in fruits and vegetables also we can try, because the concentration of in the fungicide is also very much. The use of fungicide, you know that it's a very high in case of potato, to control lead blight of potato, tomato. So we need to use or popularize this bioagent against this leaf spots. Apart from that, the mildews, where farmers or we don't know that which, for the farmers they don't know powdery mildew, they may spray a fungicide, but what happens, it may increase the incidence of downy mildew, because we know that suppose downy mildew, first if I am taking the example, for managing a downy mildew we can spray metalaxyl plus mancozeb, but metalaxyl is not effective against ericitic. So what will happen, the incidence of powdery mildew will be there. So it becomes very difficult for farmers to manage these diseases by single use of fungicides, and we are actually creating another disease. So actually we tell it iatrogenic disease. That is a disease which occurred due to prescription of a doctor. So these diseases are occurring due to wrong prescription or a prescription of a doctor for the chemicals. So to prevent that, we should promote the trichoderma. So which will be effective for both, as well as the powdery mildew as well as the downy mildew fungi. Then for wilting, I have very interesting some this finding
Yeah.
in banana.
Do the little mistake and I'm Yeah.
Banana, in banana, we have observed that banana we have two diseases. One is your this Panama wilt, and another one is moco. So both moco is caused by bacteria and Panama wilt is caused by fungi. So farmers again find it very much difficult to control them by use of, first so they have to identify it. They don't know which one is which one. Even extension functionaries, they don't know. Even scientists get confused about to identify the wilting. Is it caused by bacteria? Is it caused by fungi in field condition? You will not get so much time to go for molecular identification for prescribing in the field. So in these cases, we can promote this trichoderma, which will be effective for both for bacteria as well as for fungal in field condition. So ICR is have one product for management of, they have already done some work for managing this wilting in banana, but still indigenous and potential bioagent need to be identified, because this is the real problem of farmers where the plants are being damaged. So I have found that even in some areas, the bioagent which has been prescribed by some agencies are not working. So the duty is with us to identify the potential bioagents, to work with them and to manage these pathogens. Then in mango, tuberous, and flowers also we can popularize the trichoderma. We often found that flowers are being affected by some diseases, post harvest diseases. We found that when we are bringing them, they are being infected by botrytis. So there can be, we can work about to spray spore suspension of trichoderma on those flowers to prevent the attack of botrytis. So there are lots of work, lots of opportunities to for the to manage the disease, or we have lots of work to do. Then if you will go for formulations, so there are different types of formulations of trichoderma, but if we can divide them basically in three: solid, liquid, and encapsulated. Some ICR institute from South India, mainly from that ICR Calicut, that spices research. So they have done some encapsulated things. Liquid formulations are also now being done, but your majority you will find in the market are powders only. So talc is the dominating one, where we are finding the we will find it in markets. So nothing you have to, what you have to do, you have to isolate the pathogen. You have to go for serial dilution. So we will have this one. We'll see. So maybe this talc formulation. So here we are also in Tripura. We have done this talc formulations also. Only we are growing this after actually isolating, after bioefficacy test of the bioagent. You have to mass culture them in potato dextrose agar growth, and then you have to harvest the mycelium and you have to mix it with talc powder. So the ratio will be 1:1, and you have to add CMC, carboxymethyl cellulose. So then that thing is there. Then you have to check its spore, this one count. The CFU count, the colony forming unit, it should be 2 into 10 to the power 2, as per the standard operating procedures guided by Government of India. So there can be a different delivery techniques for this fungal antagonist, mainly trichoderma. So the majority which we are using in our agriculture is seed treatment and soil treatment. Seedling dips is also, farmers are less using. Soil drench is may not be that much effective in areas where there is high temperature, or if the soil is very sandy. And if you will give after planting, if you are drenching with the soil with the trichoderma, then they may get exposed to high heat and it may cause some problems. Then fluid drill technology, where pre-germinated seeds are being exposed with Trichoderma propagules, and it has been has been used in soil by a specialized seed drills. So we will go the important one we will discuss for seed treatment. Actually, somewhere you will find 4 gram per kg, somewhere you will find 10 gram. So you can use based on crop to crop. But and because we know that in a creep system, there are trials in agriculture, and the standard dose are being advised. So 5 to 10 gram of this talc formulation can be used per kg of seed, and the seeds are to be taken in a clean plastic container, and we have to sprinkle the seeds with some water. Then talc formulation needs to be sprayed, and we have to mix it thoroughly, and then allow for shade drying. Don't expose it in sunlight, and then we may go for sowing. So seed treatment, we, although we tell that we are doing 100% seed treatment, but in field conditions we have observed that farmers are still not going for seed treatment. So we all need to sensitize them for seed treatment. Then seedling dip, where the mainly for vegetable crops and in banana culms and all, 10 gram of talc formulations can be, we can mix it with 1 liter of water, and then we can dip the seedlings for 2 to 3 minutes. Then we can go for transplanting. So seedling, as we have already discussed, that what will happen, the roots will be colonized by the trichoderma, and this colonized root will give induced resistance, and the plant will show this plants will show resistance. So like that. Then for soil treatment, this is again a very interesting thing. That one need to be mostly, we need to popularize that we have to add the trichoderma with, actually we can add fully decomposed farmyard manure or cow dung manure. Preferably, it's my experience that the a cow should not, we should not feed a cow with uh, the grass which have the seeds, because if the seed is there, the cow dung which we will use, it will have it may have some seeds which may germinate, and it may cause some problems of weeds. So it's a better to have cow dung which we have from a cow who has eaten only a green fleshy leaves. And we can also use neem cake, we can use mustard cake, and we can use vermicompost. We can make them in a different parts. Then we can add them with your trichoderma. We have to mix it. The preparation should be like that, that if we are making a ball, it will not fall. It will we can make a ball of that formula, that mixture. Then we have to keep the thing in a shady area to allow the growth of the organism many folds. It will increase the growth of the trichoderma to many folds. So it's just like when we make a curd every day in morning, we are taking a bit of spoon of curd, we will add it in a milk, and the whole milk turns to curd. So it's like that. We have to take some trichoderma, add it with bioagent, this farmyard manure or compost or neem cake or mustard cake. Then the whole thing will become a trichoderma enriched compost, so that we can use in the field to manage all these soil borne pathogens like rhizotonia, fusarium, Ralstonia, if it's a bacteria, and others. So here we are showing some mixtures. You can see the maybe it's your mustard cake. It seems to be mustard cake, and the green sporulations of trichoderma is being easily observed. So these are some enrichment part which they are doing in the field. So what the status of research, actually we in Northeast India, we don't have that much big facilities, but we have little bit what we have done. We have isolated different trichoderma species from different regions of Tripura. The characterization we have done. One isolate we have also registered in National Facility for Culture Collection at Agarkar Research Institute, and accession number has been obtained. We have also studied in vitro efficacy of this trichoderma against different pathogens like Sclerotium bronchi, Fusarium, and pot experiments. We have all these things we have done in our college, and we have identified some native isolates against this trichoderma. The main thing before, actually our main target was to promote it to farmers. So we have actually done some trials. I will show you in this cowpea plots. Just one lab. This is the control plot where there is, we have grown these cowpea plants, and what happened on the basal part, we have inoculated it with Sclerotium ralsii. Here, basal part we have inoculated with the pathogen Sclerotium ralsii. And here both after inoculating Sclerotium rhombi, we had added Trichoderma. So what happened? This Trichoderma has taken over the Sclerotium rhombi, and all the 100% actually, yeah, there was healthy plot where healthy potted plants where we have added Trichoderma, and all the control plant, because Sclerotium rhombi is a saprophytic pathogen. It is a huge enzymes, means a huge potentiality of producing enzymes and toxins. So it has killed all the plants. So this result was the first result where I actually personally got interest and confidence that Trichoderma will work. Because we need to have a confidence also before giving it to farmer. I request all these researchers that we need to have a very good. We can put some, apart from doing RBD and SPD, we can check the things in a pot in our with our eyes, so that we will get a confidence in our product. So this is the modified Trichoderma selective media. Although you can grow the Trichoderma on potato, dextrose, agar media also, it comes. And you have to go for soil dilution, and then the colonies will come. If in first 3 days, it will be whitish in appearance. Then slowly, it will turn into olive green. So these are actually interaction where the bioefficacy test. What we have done, we have isolated 10 Sclerotium rhombi from different plants here or different sources like elephant foot yam, soil, chili, tomato, tuberose, and we have seen the interaction between them by it's like a dual culture. So we have kept them, and you can see the reactions are different. Here this Trichoderma in tuberose, it is dominating, while in Chile, there are some, the growth of the isolator of the Sclerotia prolsa in Chile is also very high. So it is trying to compete with Trichoderma. So it shows that the not be reaction will always not be actually same. So it may get different from isolates to isolates. So the reactions can be different. The all isolates of pathogen may not have same type of reaction with Trichoderma. So this is the thing, like brinjal, the other crops which we have isolated were brinjal, barley, potato, groundnut, and cumin. So from them also the reactions we are being shown here. So the reactions will be different. Here, in some instance, actually, interestingly, the trichoderma was not that much effective. That is, Sclerotium rholsi was able to grow on these things. So we need to. This is the reason that we need to isolate. We need to test the bioagent not only with some pathogen, with different isolates of pathogen, to come to the conclusion that my bioagent will be able to have a broad host range and will be very much effective in field conditions. So here also a few years back in a project given by ICR NC we have isolated some these five sources from five potential trichoderma this isolates. So they were different in their cultural growth, and these are the, see, you can see there here the sporulation, the ring type is there. So different things comes. Sometimes some yellowness also can come. So this was the some dual culture with Sclerotium rhamsi. So now this one we have all I have already discussed. So one paper actually we have published in MDPI. There we have done a little bit management of soft rot of ginger. Ginger is it is an important crop in Northeast India, and here damping off is there, or soft rot is also there. Soft rot by Pythium. So we have used this trichoderma for managing the disease. So we have found that it was effective like other biocontrol agents, as well as it was effective like your copper oxychloride against this Pythium epinidermata. The percent mycelial inhibition growth was 90% at 1500 ppm of COC, and it was 94.2 when we have used copper oxychloride at 2000 ppm. The same thing actually we have observed that in the trichoderma is effective. But here the interesting thing is, if we can see the right hand side, this picture, you can observe that now trichoderma, this was the inhibition zone. But see, now the sporulations are also there. In this part also sporulations are there. The mycelium, which is seen white, if we will take the mycelium and we will grow it, subculture it, we will find only trichoderma. So actually percentage inhibition theory, the percentage inhibition does not work in case of trichoderma. Because trichoderma, it will coil it, and there will be trichoderma. Trichoderma may be killing this white one after 7 or 10, 11, 12 days also. So a time will come when the whole this thing will become green, or there will be green sporulations on this white mycelial growth of the pathogen Sclerotium. So if we are taking up a bit of mycelium from that white part also, we will get the trichoderma. So this is the interesting part. So in a naked eye, it is not visible, but when we will see it in microscope, we will find the trichoderma, mycelium, or even though if we are subculturing it, we will get only trichoderma. So yeah, this is the same as a proof. This one, one bioagent, one side was bioagent, one side was your pathogen. But after 14 or 21 days, the plate will become like that. So it shows that trichoderma will growth 100% growth. It will overcome the pathogen 100%. So another I don't know how much time I have, but I will try to make it short. This was an again a good interesting story. In our we have done us we have an important problem of collar rot in our in pigeon pea. So this is the symptom. The pigeon pea plants, when we will grow them after 14 or 21 days, they start showing the symptom of collar rot. So if we will remove these plants, then we will find this fungal mycelium sclerotium rolfsi. Average in disease incidence for this was around 8.5%. So what we have observed that we have added a trichoderma mixed enriched vermicompost in these plots, and there was after 1 or 2 years there were no the incidence of this collar rot was very very minimum, only 1 to 2%. So the same thing happens for Phytophthora as well as for foliar blight caused by rhizoctonia. So this is again a new type of disease which we have observed in our ECRIP program of pigeon pea. Where this we have reported this one. One scientist from Africa has reported, and we have reported from India the Rhizoctonia solani causing foliar blight in pigeon pea. No more these literatures you will find in classical books if you search in internet. So we have recorded this type of problem. We have found sclerotia in this foliar blight, and here also we have seen the spraying of trichoderma or incorporating trichoderma enriched vermicompost or neem cake and enriched trichoderma enriched mustard cake. We will find the incidence and severity of rhizoctonia solani will decrease. So after getting this type of exposure, we have included the program of trichoderma in our ready. We have started mass culturing. So here, as they are this picture, they are preparing the media. The pathogen was isolated. These are the potato dextrose broth. We have extracted the trichoderma So all those things, and then this is the mixture of trichoderma with some compost, and then the product in talc powder also we have done. The picture has not been here shown. Then we have distributed it to various extension functionaries as well as farmers. And here also there is another second batch where we have sensitized it among farmers and we have distributed. This is the in the potato dextrose growth, the growth of the trichoderma. And we in Tripura, we have also disseminated it among farmers. These are the actually native isolates of Tripura. The trichoderma was isolated from our soil and it was used against the pathogens of available in Tripura. So we have popularized this trichoderma in our agroecological system under a project given by Department of Science and Technology. So he was the person of department from the Department of Science and Technology, and he's the principal of our College of Agriculture Tripura. Uh, Myself with the farmer, and now now if we will think that in respect with Northeast, we know that there are work done by A.K. Pandey, that he has done a notable, he has isolated a Trichoderma viridi strain, which is very much effective for tea gardens. And when it has been applied in the rhizosphere of tea gardens in Assam, it acts as a broad spectrum antagonist against tea plant pathogens, mainly Pastalocia thiae, which causes leaf spot and leaf blight of tea, as well as Fusarium solani, causing root rot in tree. Through the actual mechanism, what he observed was maybe due to mycoparasitism and extracellular enzyme secretion. Like in Manipur also, there was report that some indigenous trichoderma isolates were found to be strongly inhibitory efficacy they have shown against Pythium epidermatum. The work was done by Kamla and Indira Devi in the year 2012. This one, I have included that the persons, maybe apart from Northeast, also in Northeast, maybe if someone is from West Bengal or in, I don't know, in Odisha, betel vine is grown or not, we should promote the growth of the use of trichoderma in betel vine plantations, as the leaf here is directly consumed by us. So we should discourage any use of fungicides or chemicals in betel vine plantations. So this is the area where we can try, we can undertake some research works. Apart from that, black pepper. So this black pepper is very much common in northeast areas as well in southern parts of our India. So yeah, I know that in Karnataka or in Tamil Nadu, Kerala, these parts also, the use of trichoderma for black pepper plantation is very much effective, as there are some shades also. It's a climbing type tree and shade is there, so heat is not that much, and the growth of the trichoderma is very optimum. It has a good environment. It gets a good environment to act against pathogens. Apart from that, now we are also promoting pineapples in any India, Tripura. We are promoting queen pineapple. So we can, in these hilly areas, actually, upland areas, any use of any fungicides, with the help of rainwater, they will go into lungas. So then again, they will cause a havoc. So in this respect, we should promote use of trichoderma in upland situations, mainly in the shifting area, shifting the areas where shifting cultivation or jhum cultivation is also being practiced in northeast area. Apart from that, if we will if we will not, if we will exclude jhum cultivation in upland areas also, we have to promote bioagents in forests. We have to promote bioagents, because if we will promote any chemicals, we know that it will cause damage to ecosystem very harshly, and the biodiversity of the ecosystem will be damaged. So we have to keep the biodiversity of the ecosystem intact. The DBT NACAP from Assam, they have done some novel work. They have isolated microbial strains which are effective for northeast areas. So why any India is important? Sir was also telling about the northeast area because it has the high biological diversity. So bioprospecting native microbial strain from these areas need to be identified. Large areas are there which have potential for here. The mainly we agriculture we are using very low inputs of chemicals, and more areas are under organic. So the opportunities are very good. Then the conditions here is high rainfall and humid conditions, which favors many fungal diseases, as well as it will favor the growth of biogens also. Because the place where actually there will be deer, there only the tiger will be there. Without deers, tiger can't thrive. So if there are fungal pathogens, then only the trichoderma will be there. So crops we have in northeast this rice, vegetable, ginger, turmeric. So the plantations crops that can be benefited by use of biological disease management like trichoderma. So recent ICR work has been explored, and it has been found that there are some strains. This is just an information that they can survive for 1 year under room temperature. Trichoderma asperellum. So ICR NRIPM they have developed it. So I am just showing it to sensitize the young research scientists to initiate works on trichoderma or any other bioagents or biofertilizers. So in future, if you will go for future research scopes. So yeah, molecular identification and characterization of elite strains needs to be done. Metagenomics needs to be studied. Indigenous trichoderma strains needs to be discovered. And in formulations, we have to do more work in encapsulate to more popularize this liquid and encapsulated formulations. Microbial consortia, definitely some work has been done in Northeast India as well as in different parts like in ICR, IIPR also to develop consortia of trichoderma with pseudomonas. But more interventions, more innovations needs to be undertaken from in this area. So these are the some references which I have used, although I have kept my slide to limited one, but maybe I have taken more than 1 hour. So thank you for listening me patiently, to all the scientists, students, research workers. This is the picture of my College of Agriculture Tripura, and this is the goddess Tripureshwari from Tripura. So I invite all of you to visit Tripura and to have blessings of the goddess Tripureshwari. So thank you.
Thank you, Avasti sir, for this was a very nice kind of informations you have provided on there. So now we have some questions, sir, asked by some of our dignitaries. So I go one by one. So one of the persons, Susma Sudarsgaran. So she asks about what are the research gaps is you already mentioned several extensive studies. I must say that you also addressed few about recent and what kind of on there, but still, if you want to be addressed more on this, so for future research, what could be behind on there, please, sir.
Sir, future research thrust for future research.
No, they are asking, since there are several research done, what kind of research gap is still behinding on to work with trichoderma?
Sir, may I get this? They are telling that there are
There are so much research already been be done with trichoderma.
trichoderma. Okay, okay,
So what
okay,
kind of research gap is been be? To my understanding, one of them is
okay. What kind of research gap is there to what? For popularizing it?
no to do more research with trichoderma.
Okay, okay. 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 So I think actually there are lots of works have been done in trichoderma, but the science is not static. Every day, due to climate change, actually new pathogens are coming up. The diseases which were minor previously are becoming a major one nowadays. The gap is actually, there is not gap, there is a need of continuous research. The basics always needs to be ready. We can't think that only this having I have reached a potential trichoderma, now there is no need to do research, because that potential strain will not be that much effective. It may not be that much effective after few years. It is just like fungicides which develop resistance. So research, we need to have more research towards developing those type of strains which will be climate resilient and which will adapt like a pathogens. The young scientists, they can think then that you can develop, you can use genetical engineering and you can make them the strains which will be more adaptable to new kinds of environment, to the new paradigm shift which we are facing in agricultural science. So they will be more adaptable to that side. So yeah, I think I have answered it.
Yes, sir. Then next, Udayo Bhanusali say again asks, can we work upon synergetic effects of fungicides and biocontrol agents? She
Very good, very
primarily
good,
works on
very good question.
Mancozeb with trichoderma against FOL. I think FOL means
Yes.
Fusarium oxysporum is telling.
Actually, yeah, yeah, very nice question. Yeah, as we know, for fungicides, let me tell about some first for bio pesticide. Like for mixing trichoderma with pseudomonas consortia, we know we are developing. But if we are telling about fungicide, trichoderma is compatible with some fungicides. It's there. Although fungicides means what? A chemical which will kill fungus. But interestingly, many research work has been done, and some research scientists have reported that this trichoderma is compatible with this fungicide. It means that if we are using the fungicide, then also this strain will not be killed 100%. The population of that strain, if it will be diminuted to 40% or like that, or 60%, and then again, because it's a living organism, it will again sporulate, it will again produce conidia, and it will again work on pathogens. So it is there. There are some research works in this. Some research scientists, they have already reported compatibility. They have already studied compatibility of trichoderma with fungicides. But yes, before testing, we should not recommend any fungicides, because we know fungicides are harmful for antagonist like trichoderma. But yeah, after doing research, very good question. You can search for strains which are very much compatible with fungicides, and you can promote them. So means after doing registration and all, so research can be done. I think very nice, good idea.
Yes, sir. Then another question from her is, what are the guidelines for solid formulations with respect to viable spore slash gram of solid formulation?
Yeah, yeah. Actually, you need to just you have to search in Directorate of Plant Protection and Quarantine Storage website. You will go there. The standard SOPs are given. So you have to search only the standard operating procedure with the guidelines for production of trichoderma. The government of India in the website. Directorate of Plant Protection and Quarantine Storage has given all the guidelines of this the quality as well as the procedures, all those things to have this biocontrol agent.
Yeah, then a student is asking, do you have or your institute have to do intensive? Do you have facility? Do you want to be
Uh,
answered?
internship facility, actually, yeah, you have to write to our dean. If he agreed, then we can have. But actually, your institute also have to be agree. Then we can go. We have to talk. Yeah, it's a good thing. We can think of that.
Yeah, then Dr. Monica Sharma asking that have you tried this fungi against stress like salinity tolerance?
No, no, no. I have not tried it because we don't have saline soils here. This is a little bit actually. It's around 100 kilometer. It's in Bangladesh. We will find saline soils. So no, when the partition was done, northeast is in hilly areas. So around 60, 80, or 100 kilometers only we will get soils nearer to seashore. So I know in Odisha you have been surrounded with a vast area of sea and saline soils are there. I was previously in OUT, so I have done some work in Odisha also in forage crops. So it is very much interesting. You can visit in beaches. You will find their soils. Maybe first you may get failure, but maybe you may get in first chance success also. Who knows? And maybe after 2 or 3 attempts, you may get some nice strain of trichoderma, which will be a soil tolerant to soil salinity. But in respect with India, lots of work has been done, and ICR must have the soil tolerant species of trichoderma. But as I told, the biology is changing day by day. Having one does not mean that it will is okay. Pathogens are changing. So we should have, as we know, the missiles are changing, drones are changing. So we should have a good, better drone to resist our land with the enemy drone. So like that, you should have always a better potential native strains to protect your crop from changing and virulent pathogenic strains. So who will do? Yeah, microbiologists. You people have to do, young minds, the researchers, they have to do. So please carry on with this good work. Very good question. So I hope that in future you will come up with a very good potential saline, salt tolerant trichoderma. Very good question.
Okay, sir. Then from Sriya Pharmex Solutions, AJ Cons, they are asking, self-life of trichoderma can be critical challenge for adaptation. So what could be the best possible method to overcome this challenge?
One can be its genetical makeup, as I have already shown that one strain of Trichoderma asperellum can withstand 1 year in a room temperature. So the first thing is in among this variability, we have to isolate the organism. That second thing is encapsulation or providing them like in encapsulated trichodermas or providing them an environment which will save them from harsh environment. And the third is means they should be conditioned and providing them with substrates. So when we are providing them with substrates, then the survivability chances will be more. So apart from that, I think that's not a bad if you can have 1 year or 6 months even the shelf life, then it's not a problem. Yeah, because it's not a after all it's not an iron that we should have a shelf life of 2 year, 3 year, 4 year. 6 months is a good one.
Yeah, okay. So another is that from Kalini Sinde want to be know if there is a higher dose of trichoderma in the field, could it be toxic or harmful to the crop?
No, 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 it won't be toxic. It won't be toxic. It's a, like a Ayurvedic, or it's like actually anything is harmful in excessive dose, but in soil it's not harmful because we know that how much you will be add. Soil is very, very vast thing. It won't change the soil dynamics so easily. So whatever dose you want to provide, you can give, but please add it with some substrate. Otherwise, it may not survive very nice. So when we are going for soil amendment, add it with substrate, the more and more trichoderma, because you see it will degrade, it will inhibit the pathogenic organism, and it will induce resistance. So the plants will be healthy. So there is no problem.
Okay, very nice, sir. Then another, will it vary with respect to geographic and climatic conditions, strain variants? So, okay, you
Yeah,
already
definitely, sir.
answered.
Definitely. Not only, yeah, definitely it varies from geographical and the areas where there is a warm temperature, high temperature, the strain of that area, the trichoderma is definitely different from the areas where there are cold temperature. So that's why survey is required. Survey, regular survey and collection of samples and isolation is required. Then their compatibility check, bioefficacy checks are required.
Well, another question is given by Dr. Mahajans is asking about trichoderma effective against sclerotina office inhibition. So she is more interested, but you already show some of those in your slides. So
Yes.
if you want to be add to, okay, how it is effective towards sclerotina rolfos, you can say also.
As we have already discussed, the mycoparasitism. So it plays a first role as a daughter plant. I have shown that it will coil the pythium, that is Sclerotium broadside, then it will draw Hostoria, it will derive the nutrition. So it's known as mycoparasitism. Apart from that, antibiotics will be there, where some biochemical substances like trichodermine will be secreted. So it will also kill the we can easily observe in microscope. If we want to see it in our eyes, what we can do, just keep this in dual plate culture and try to, from the interaction center, try to where the trichoderma, from the interaction center part, just take the mycelium and observe it in microscope. We will find that antagonistic organism is coiling the hypha of trichoderma, the sclerotium.
Yeah, but she
So
is more
seeing is
interested
believing. We
towards,
will be, yeah,
yes,
seeing
sir.
is believing. We will be easily this one. हम मान जाएंगे. And the second thing is in soil, what happens? The sclerotium outside, the sclerotians, they survive in soil. Sclerotia. So what happens? The trichoderma will attack, infect the sclerotias, and they will colonize the sclerotias. Sclerotias will not germinate after the colonization.
Okay, sir. She is more interested towards knowing about absolute value of some percentage of inhibition or EC50 if it's calculated.
Actually, yeah. Here, actually, we ED or EC, not ECOED, we have to see about Bell scale. If in internet or in paper,
Okay.
we will search, we will get Bell scale reading. Because when we are using with some chemicals, we may find actually percentage. Here also we can find it percentage inhibition if you will grow in dual culture technique both the organisms, but that is actually not the justice. Because after there can be some inhibition zone, but as I told, the trichoderma will advance and it will coil the pathogen. So if we will keep the plates, so there we will find that there will be 75% antagonism by the trichoderma, or the sclerotium rholsi will be 100%, or any pathogen, taste pathogen may get 100% covered by trichoderma. So to know that, we have one Bell scale reading. So please go through with that one, or you can have, if you find any difficulties, you can text me or email me. I will help you. Then by following Bell scale reading, you can work and you can know about bioefficacy of trichoderma and you can publish papers.
Yes, sir. The last question by Dr. Thangamani. So, is telling about any substances which can be added to your soil for the with the trichoderma applications.
Yeah. As I have already discussed, neem cake,
Yes.
vermicompost, farmyard manure, and then mustard cake, they can be mixed with trichoderma and then we can incorporate them in field. First, we have to mix them 1 kg. We can take trichoderma, 100 kg. We can take this organic manures. We have to mix it. We have to incubate it for 21 days as per prescription of different ICR institutes like ICR, IIHR has also done it. But in field conditions, farmers does not go for 21 days incubation. We can go for 7, 14 or 21, but 21 is best. And then we can incorporate this mixture into the soil to get management of the soil-borne pathogens.
Okay, thank you, sir. Before I give you a formal vote of thanks, I feel like our founder president, Professor Dr. Arvind Desmukh sir has joined, and I really want to request him to address to the gathering. Dr. Desmukh sir.
Thank you. Thank you very much.
Thank you, sir. Thank
So
you, sir.
I was watching a lecture of the sir, Avasthi sir,
Sir.
and the topic is was a very excellent. It is a need of the nation. Whenever many people are asking which should be the organism of the nation, I will be. I'm a fan of Trichoderma.
Sir.
Trichoderma is a really friend of farmer,
Yes,
and
sir. Yes.
yes, and Dr. Avasthi has focused light in detail on the trichoderma. I also appreciate the efforts of the chairman of Biofertilizer Awareness Committee, Dr. Sujok sir. He is continuously arranging various lectures on the biofertilizers and biopesticide. Now you see, we are using chemical fertilizer, chemical pesticide because we want more money. But ultimately, we are spending money for our health, the health hazards caused by chemical fertilizer and chemical pesticides. So this is a late, not right time. I will not tell right time. We are late in adopting biofertilizers and biopesticide. So today's lecture is having a great contribution in creating an awareness. We have recorded this lecture, and tomorrow it will be again circulated. Recorded version will be circulated in all groups. As a president of Microbiologist Society, I appreciate Dr. Sujok and I thanks Dr. Dikshit for delivering good lecture. I also thanks all attendees. I was watching many PhDs, many doctors, and many students have attended this lecture. Thank you. Thank you very much.
Thank you so much, sir.
Thank you,
Thank
sir.
you for So, Dr. Awasthi, it is really a very nice kind of, you have started from the importance of agricultures, about the biology of trichoderma, all kinds of other fungal infection, their pathogenesis, their morphology. Then you come with the compound alpha pyron derivatives, then about seedings, about horticulture, all different importance about of many formulations of torque, seedling diff. So those are how it will be compatible with organic manure, vermicomposts, different types of cakes, neem, mustards, etc. Then you went for the field trials on your own studies, giving lot of examples with coffees and all this, pineapples, black peppers. So many of very important diseases of coloroids and all this. So this is a perfect example of how you are being be able as a scientist for ICR to back all those lab work from field to farm and to the farmers. So this is what the nations wants. So I really thank you so much from my heart of hearts, from all the members of Microbiology Society of India, the hostings, my colleagues, Dr. Sivani Mahapatra, our president, Dr. Desmukh, all of them. So we are really grateful to you, sir. It is really a very informative informations. And you have also explained about all the questions asked by our participants. So it is really very nice interactions. We are really grateful for that. So thank you all. And I wish all you have the best of success in your life. And we are now closing these sessions, and the recording versions will be shared to all of you, those who have not also attended. Thank you all of for your participants. And we just now to we take a photographs. So if all this those who have could be on switch on your camera, then we will take some of the snapshots and then we will be add.
Thank you, sir. Thank you to the organizers and to the Microbiology Society, sir. Thank you.
And Dr. Avasthi, sir, can you please switch off the slide sharing mode?
Light switch off.
The slide sharing mode.
She's asking for to stop the slide sharing mode so that all photographs can come.
Okay. Thank you. Just a minute.
Okay, thank you, Dr. Sivani. We'll stop now.
Yes, sir. Yes.
Prathamesh, you can now close.